There's a pill. You take it every morning, every night, for the rest of your life. 30,000 doses if you live to 80. Skip too many, and your arteries quietly fill with plaque. Heart disease is still the #1 killer of human beings on Earth. 19 million deaths a year, 1 in 3. Now imagine a single shot in your arm. 1 hour, done. It rewrites a single letter of your DNA, and the gene that has been pumping cholesterol into your blood for 50 years just turns off forever. That sounds like marketing hype. It sounds like science fiction. But on April 14th, 2025, 14 people had it done. Their LDL cholesterol dropped by an average of 53%. One patient dropped 69. This is the story of how that happened, and it almost didn't. Heart disease has been humanity's biggest killer for as long as we've been counting. In the United States, someone dies from it every 34 seconds. By 2050, the number of cardiovascular deaths could hit 35 million a year worldwide. We do have a weapon. It's called a statin, lovastatin, atorvastatin, rosuvastatin. They cost pennies. They work. But here's the brutal truth. 1 in 3 patients stops taking their statin within the first year. They forget. They feel fine. Life gets busy. And for people born with familial hypercholesterolemia, statins are often not enough. Their LDL stays dangerously high, no matter what they swallow. For these patients, the pill bottle is a daily reminder of mortality, a small plastic countdown. What if you could replace that reminder with a single moment? 1 injection, then nothing. That is the dream. And in 2025, the dream walked into a clinic. Here is what most people think when they hear CRISPR cures cholesterol. 1. It's science fiction, decades away. 2. It will replace statins for everyone next year. 3. It edits your sperm and eggs, so it changes your children too. All 3 are wrong. The truth is more interesting than any of them. It happened. It is real. It is in living humans right now. And it is also more limited, more expensive, and more permanent than the headlines suggest. To understand why, we need to go back to a small Texas city in 2003 to a doctor who asked the inverse question. Not, who has too much cholesterol, but who by accident of genetics has almost none. In 2003, at UT Southwestern in Dallas, a geneticist named Helen Hobbs was running something called the Dallas Heart Study. 6,000 adults, all ethnicities. Decades of blood samples and follow-up. A French team had just linked a gene called PCSK9 to a family with terrifyingly high cholesterol. Hobbs flipped the question. If too much PCSK9 makes you sick, what about people whose PCSK9 is broken? She looked, and she found them. About 2% of African Americans in her cohort carried inactivating PCSK9 mutations. Their LDL cholesterol was naturally low. Their hearts were quietly, statistically, dramatically protected, even when they smoked, even with diabetes, even with high blood pressure. Their heart attack risk was reduced by 88%. Hobbs had found people who were, in a real sense, born with the cure. PCSK9 is a protein that grabs those vacuum cleaners and drags them inside the cell to be destroyed. More PCSK9, fewer vacuum cleaners, more cholesterol in your blood. Less PCSK9, more vacuum cleaners, less cholesterol. The 2% of people with broken PCSK9 had, by sheer luck of birth, been given a permanent upgrade. What if we could give that upgrade to everyone else? To do that, you needed a tool that did not exist. In 2012, Jennifer Doudna and Emmanuelle Charpentier published a paper that won them the Nobel Prize. They showed that a bacterial defense system, CRISPR-Cas9, could be programmed to cut any DNA sequence you pointed it at. It was molecular scissors. Astonishing, but messy. CRISPR cuts both strands of the double helix. The cell tries to repair the cut, and the repair is unpredictable. Then, in 2016, a chemist at the Broad Institute named David Liu had a quieter, deeper idea. What if you didn't have to cut at all? Liu fused a partially deactivated Cas9 with another enzyme called a deaminase. The result was a base editor, a molecular pen. It could swap one DNA letter for another, A to G, C to T, without breaking the helix. Without the messy repair, it was the difference between scissors and a typewriter correction key. Liu won the 2025 Breakthrough Prize for this. As of 2026, at least 23 clinical trials use his base editor or its successor, the prime editor. The molecular pen was real. The protective mutation was known. All someone had to do was put them together. In 2018, a Boston cardiologist named Sekar Kathiresan founded a company called Verve Therapeutics. Kathiresan was not a stranger to heart disease. His own brother and other family members had suffered early heart attacks. He had spent his career studying the genetic causes of the disease that haunted his family. His vision was simple and audacious. Take the protective mutation that nature handed 2% of people. Engineer it into anyone who needs it. With a single shot, use the LNP technology that delivered the COVID mRNA vaccines as the wrapper. Use Liu's base editor as the cargo. Use a guide RNA to point it at exactly one spot in the PCSK9 gene. Make a permanent edit. Turn the gene off. Walk away. In July 2022, the first human in history received this drug. His name has not been released. He was an adult with familial hypercholesterolemia who had already survived a heart attack. The trial site was in New Zealand. The drug was called Verve 1010. For 18 months, the data looked beautiful. Up to 55% LDL reduction. Durable through 6 months. The world started to whisper that this might really work. A patient at the second highest dose developed 2 warning signs within 4 days. Liver-enzyme spike, vein grade 3. Low platelets, grade 3. Both transient, both fully resolved. No bleeding, no lasting harm. But the numbers were serious enough that Verve paused. Enrollment immediately. If you were watching from outside the field, the obvious conclusion was that gene editing was simply too dangerous. That the body would not tolerate having its DNA rewritten on a hospital cot. Inside Verve, the engineers asked a sharper question. Was it the editor, or was it the wrapper? The base editor itself worked. The mRNA was clean. The guide RNA was precise. But the LNP, the lipid nanoparticle that carried the cargo, was a more conventional design. It activated the immune system in ways the team had not fully predicted. So they did not abandon the program. They redesigned the wrapper. The team kept the lesson, threw away the wrapper, and started over. The new drug had a new name. Verve 102. May 7th, 2024. The first patient received Verve 102 in the United Kingdom. Then more patients, Canada, Australia, Israel, New Zealand, 3-dose cohorts, 14 people, all with severe inherited high cholesterol, all with at least 28 days of follow-up. April 14th, 2025. Verve announced the data. At the highest dose, mean LDL cholesterol dropped 53%. The peak in one patient was 69%. PCSK9 protein in the blood fell by 60%. No serious adverse events. No clinically significant lab abnormalities. One mild infusion reaction, treated with acetaminophen. The comeback worked. 3 days earlier, the FDA had granted Fast Track designation. 2 months later, on June 17, 2025, Eli Lilly announced it would acquire Verve for up to $1.3 billion. Sekar Kathiresan moved from Verve CEO to Senior Vice President at Lilly. The trial only enrolls patients with severe genetic cholesterol disease, not your uncle who eats too many burgers. Long-term safety past a few months is unknown. The edit is irreversible. If something goes wrong in 15 years, you cannot undo it. A statin costs pennies. A one-shot CRISPR therapy will likely cost between $300,000 and $1 million upfront. Broader use? Beyond familial hypercholesterolemia is 5 to 10 years away. So, pause for a moment. Sit with this question. Would you let a doctor edit your DNA permanently in exchange for never taking a cholesterol pill again, knowing it cannot be undone, knowing the long-term unknowns, knowing you would carry that edit for the rest of your life? For some people, the answer is obvious. For others, it is uncomfortable. There is no clean script for this kind of decision yet. A statin can be stopped. A CRISPR edit cannot. The first generation of patients who say yes are saying yes for everyone who comes after them. But what fascinates me is the quiet shift underneath the question. Medicine is becoming something you had done, instead of something you take. The pill bottle on the counter is being replaced by a single moment in a clinic chair. That moment is small. The DNA letter is microscopic, but the change reaches forward across decades of a single human life. 6 months after his infusion, that first patient walks out of his cardiologist's office. He holds a lab printout. LDL 78. He folds it into his pocket the way other patients fold prescriptions. Same hands, new future. One letter of DNA, one injection, one quiet rewrite of a 50-year story. Think about what just happened in this video. We followed a single edit, in a single gene, in a single organ, and we watched it ripple out into the entire architecture of cardiovascular medicine. A 2% accident of human genetics, copied through chemistry, delivered through a fatty bubble, written into the liver of 14 volunteers. And now, potentially the future of how 1 billion people live and die. That is the real wonder of biology. Tiny things, written carefully, change everything. But the same instinct that lets us rewrite a letter of our own DNA is also looking outward, pointing telescopes at the deepest sky we can reach. In the coming weeks, we will follow the James Webb Space Telescope into a result that genuinely stunned cosmologists. Distant galaxies, billions of light years away, all spinning in the same direction. Spinning, curiously, the way matter spins inside a rotating black hole. Picture it for a moment. Webb opens its golden mirror. Light that left its source before Earth existed slowly fills its sensors. And in that light, an old, almost forbidden idea wakes up, that our entire universe, every galaxy, every chalk line on every blackboard, might be sitting inside a black hole that we cannot see from within. The same scientific